David is a professor in the biology of aging at Durham University. He's also the founder of Magnitude Biosciences Limited and the chair of the British Society for Research on Aging. His background is in biochemistry and genetics in 3D models. After a PhD characterizing Drosophila P13 kinases, he switched to the nematode C. elegans. And he discovered this inhibiting folate synthesis in the bacteria in E. coli that makes C. elegans live longer. I like that. So he was asked to contribute to a paper on the heads of UK funding on the biology of aging in the UK, and the barriers to translation and to recommendation, how to recommend to overcome them. So it's very exciting as we all get older to hear about aging and how we can promote longevity and healthy living. So I'm looking very forward to listening to you and hearing what you have to say.
Right, so the British Society for Research on Aging was formed in 1939, so it's probably the world's oldest society for studying the biology of aging. It sort of started as a bunch of people interested in aging talking to each other, and in many cases that's still what we are, but we also have a charitable, we are also a charity, and we engage in supporting research and public education, and we raise money and we fund research as well ourselves, and we're trying to work with other charities and the government and so on to improve aging research. And the idea is that we want to actually do that to a point that we can actually help slow aging and make people healthier for longer. Which isn't just, which you'll see is quite difficult, you know, it's not just about doing the research. But so this is, when I was a postdoc in the Netherlands, when I came back to the UK about 20 years ago, I went to talks by, epidemiologists and economists, and they predicted that in 20 years' time there'd be a big problem to do with demographics, that they'll have an older population. So this is the population pyramid, and you see that this is from 2012, and there's a spike of people here that were in their late 60s, and now they're in their 70s. This is the baby boom. And then their children are Gen Xers, and then their children are millennials, and their children is the Gen Zs, But as these people get older, there's going to be more and more older people, and our population, our birth rate is going down. So there's more and more older people to support by less people, and that is happening all over the world. But it was predicted as a problem 20 years ago, and now we are having that problem in the UK. There's more old people that are in ill health that often retire early, can't work further. There's a huge amount of pressure on our National Health Service. There's a lot of economic pressures which are causing a lot of the political upheaval that we see around the world. I think can be traced back to this. People blame the wars and Ukraine, but it's not really. It's demographics is the problem. And inflation as well is a problem because of that. Because now there's less people to do the jobs, so you have to pay people more, and so the inflation goes. It's another contributor to inflation. So a lot of the problems that we have in the UK and many of the developed world is to do with the demographics. And so one major solution is just to make sure that people stay healthy for longer and productive for longer, and that's a way to try and solve some of those problems. So then I looked up the population, because I was coming here, I looked up the population pyramid for South Africa and tried to work out where South Africa was in there. And I worked out roughly it's about 50 years behind the UK in that. and this is the population pyramid. This is a slightly different population pyramid in 2025 in the UK. You can see there's a big bulge of people here in their 60s who are going to get, and quite a few in their 70s are getting older. And to find the shape in South Africa that would be sort of similar, you have to predict forward to 2075. Because at the moment the population pyramid in South Africa looks like this, which is a bit flatter, but you still see already that it's not as flat as it would have been 20 years before. Not as so it's already starting to become a problem, but it's not as bad as the UK is. But that means that in South Africa you have a bit more time to deal with these problems than we have in the UK. We're suffering from them now. So even if we did something about it, there's still a big problem. Not that it's going to get any better, so we still have to work on it. Okay, so, you know, for almost every disease, aging is the biggest risk factor. And when we used to think about, talk about this, everyone would think about Alzheimer's and, you know, cardiovascular disease and all that kind of thing. But actually, the pandemic showed that infectious disease is the same. You know, if you look at the number of deaths from COVID, it was hugely age dependent. I think this is in the UK. I imagine it was similar in other parts of the world. But there is very little research on aging compared to research on all the different diseases. And also aging research is difficult because it takes a long time. You need large cohort sizes, and I'll explain that a bit more. So, our Chief Medical Officer, Chris Whitty, did a report on healthy aging in the UK in 2023. And this is one of the figures out of his report. And it's comparing people from the most deprived areas in the UK to the least deprived areas in the UK. And there's a huge difference in the life expectancy compared to the least to the most deprived area. People in the least deprived areas live well into their 80s, whereas it's more like, you know, 70s for the most deprived area. But the biggest difference is the amount of time in poor health. Even though they live longer, they spend less time in poor health than in the deprived areas, where they spend 26 years in poor health on average. And you know, Chris Whitty was saying it's not to do with the meds, it's to do with the getting better medical care, because most of these people they're healthy, they don't need to see a doctor a lot of the time. So it's a quite big question of why there's that discrepancy and what we can do about it. And obviously there's socio-political questions about how you make that, but also there's biological questions, I think, about understanding that, why there's that difference. So one of the things, in the same report, there was something that came from Linda Partridge, who's a person that works on aging, a very well-known person who works on aging and part of the Royal Society. They were commissioned to talk about the biology of aging in the same report. And they came up with, which is what was termed the geroscience hypothesis quite a long time ago, 20, 30 years ago. And the geroscience hypothesis is encapsulated in this figure from that report. And the idea is that rather than working on separate diseases, you should work understanding the mechanisms of aging, and then you can slow aging, and then you can slow all those other diseases. And that's the way that we sort of proceed. And that's a lovely idea, but actually in reality, we've been talking about that for a long time, and it hasn't actually happened. You know, it hasn't been a big diversion of resources to understand how we keep people healthy for longer. We have much more resources working on different diseases. And the reason why that hasn't happened is because the world doesn't work that way. The world works within therapeutic areas. The medical profession is divided into therapeutic areas. The pharmaceutical industry is divided into therapeutic areas. And a lot of the charities that support research are, you know, Cancer Research UK or Alzheimer's, you know, all these, British Heart Foundation, all believe in particular diseases. And then, so in this report that I was asked that you mentioned, that I was asked to do, one of the things I found by talking to clinicians that much of the aging research community is not taken seriously by clinicians. So they don't understand why biology of aging is important to them. And so, you know, and then also it's very difficult to fund. So we've got to come up with a different way of doing things. So one of the ways I've been thinking about is that it comes from the observation that if you prevent one disease, you often get positive effects on other parts of the disease. So the therapeutic area that's done the best in terms of prevention is probably cardiovascular disease, where you can treat high blood pressure and high cholesterol, which in themselves are not diseases, they're just indicators, and we understand that they are risk factors for disease. So but if you treat high blood pressure, you get less dementia, or you know, if you treat, and same with cholesterol. Or there's another study where they looked at children to the immune system to looking at cardiovascular, and they found there was lower cancer rate incidence from that. So, but it kind of fits with the way that we think about aging, because aging is a network. You know, aging is about the decline of the whole network, and if you you protect one area, you can protect other areas. And I'll talk about, might get a chance anyway, depending on the time, to talk about that in a bit more detail. So within the medical profession, people are working on prevention of specific diseases, you know, and some areas more than others. But they're doing very similar things to people working on Asian research. So I think the people working on Asian research need to work more closely with people working on prevention of disease, and then work on, because if we prevent one disease, we will prevent other diseases. I'm pretty sure about that. And the things that we find in preventing one disease will be applicable to other diseases. So that's the kind of way I'm thinking about it. And I engaged a lot with cancer research, cancer prevention. And I went to, there was an international conference on cancer prevention that was organised by Cancer Research UK and the American Cancer Society. And it was really interesting, and there's a lot of stuff, and there was a lot of overlap there. People were looking at similar sort of treatments and things that people were looking at aging, doing similar things. But I was the only person really who was like an aging researcher at that conference of about 400 people. So then I felt that there needed to be more interaction. We're working now with Cancer Research UK to improve that. But even within Cancer Research UK, they have £2 million, £8 million budget, a year on prevention of cancer. But their total budget is £400 million a year. So it's only about 2% of their entire budget is going to prevention compared to treatment. But I think that's another thing that we need to look at, you know, because all diseases have these different stages when they come from an early stage. So cancer is, I've got cancer up here as an example, but this could be the same for lots of other disease states. So you know, you have early cancers, and when you have early cancer and you detect it early, you might be able to treat that, you know, take away that surgery or treat that cancer and get rid of it. But what you want to do then is prevent it going to later stage 4 cancer. So in those early stages, you are treating, but you're also preventing. You know, prevention is important. And as you go further along, prevention becomes less important, and then you just have to treat and try and, when someone's got late stage cancer, you're trying to keep them alive. Now you can say a similar analogy for rheumatoid disease or dementia or heart disease. You know, when you're starting off, you try and prevent that disease happening, but when someone's really badly affected, it's not much you can do in terms of prevention. You have to just treat them. So what I think is, you know, the way that the medical profession and the pharmaceutical industry and so on is set up is really to focus on those treatments, because there's a lot of people want to, if they're dying of cancer or something, they want to treat it, but actually use a much more better use of resources to prevent those things happening in the first place, and then trying to understand prevention. But also you have to start thinking, the problem is then you have a safety consideration, because when you want to treat a stage 4 cancer, you don't really care about the side effects, you're just trying to kill that, you're trying to stop that tumour. When you're at early stages, you can't start giving healthy people drugs that have side effects, because there's the risk-benefit analysis. So you have to start thinking about that, and maybe you look at what in the prevention of disease space, look at people with a high risk factor, high risk, you know, people in a high risk area, and then treat them, and then you have that balance is better. So there's a lot of issues about this, but I think it definitely needs to be looked at. And then in this conference that I went to, there was a really interesting talk about from the World Cancer Atlas. And this particular graph like hit home to me as someone who's, you know, based in the UK, is that this is the graph for how many radiotherapy machines there are in each country per million people. You know, so the dark colours are 5 or more, and then the light colours are less than 1 or no machine at all. So you can see in like many parts of Africa, there's basically no access to radiotherapy, and surgery as well is probably not going to happen either. So, you know, in many parts of the world, prevention is the only thing that we've got. And so that's really important, and I think as the aging demographic happens in developed countries, we're going to have the same problem, that prevention is going to be much more important than treatment, because we can't afford to treat everybody. So I think, you know, that people working on aging need to work with those working on prevention of disease, and like a lot of cross education is necessary there. Okay, so So one of the things about aging, so you might have heard of the hallmarks of aging, and some of you, who's heard of the hallmarks of aging? No one's heard of that. Okay.
I'm a pediatrician.
No, it's okay. I'm not a big fan of the hallmarks of aging, so I'm happy that you haven't heard of hallmarks of aging. But a lot of people in the aging world look at the thing called the hallmarks of aging, which is analysis of the hallmarks of cancer. You've heard of the hallmarks of cancer? People heard of that? Not really? No. Hallmarks of cancer was like, okay, there's a certain features of cancer that say that you're likely to have cancer, and we can work on those. And the hallmarks of aging is kind of a copy of that. And so this is all the different factors, and then things like, mitochondria or genomic stability, telomeres, and things that people associate with aging. But my way of looking at aging is the whole system from a very molecular level all the way to through to cell and tissue and organ and whole person and society levels as well. I think all those things affect aging. So it's a network, But there are some fundamental things about aging that I think are true. So one is that the rate of aging accelerates with time, and we see that from mortality data, that we get this called the Gompertz curve, so the mortality, so you have an exponential increase in the chance of dying, basically. So every 8 years, your chance of dying doubles. It's basically how it works. I once said this at a New Year's Eve party and it wasn't going down very well. But so that means that if you want to study aging, if you want to have this is important for like thinking about trials for aging. If you have a trial for aging in someone in their 30s and you run it for 5 years, you're not going to see much. But if you start off with someone who's 65, you're going to see much more aging happening. The other fundamental is that variation between individuals increases with time. So we see that even with our little worms, that is kind of fundamental of aging. So kind of, if you look at animals, lab animals, they all kind of develop at the same rate in the same way, but then as you let them age, they age in different ways. And that's the same for humans. So it's not just that each individual ages differently, but within an individual, different organs age differently, potentially, So that means as well that if you're doing a study of aging in humans, you need the older the people are, the larger the cohort needs to be, because you have more to account for that variation. And sometimes when people design experiments, like studies in mice, they're thinking about something that I'll be okay just using 10 mice per sample, but then actually if you're going to do aging, you need many more because of the variation that happens. And then the other thing is that you have this reduction in homeostasis with time. So the ability to deal with changes and stresses and things like that reduces with time, and that has knock-on effects as well. So like you can drink a lot when you're young and you can wake up the next day and go back to work, but when you're a bit older it's not so easy. That's an example that I like to think about, but many other things that happen. So that's basically what I've just said, that clinical trials for prevention, it's the same for prevention and aging. They just have to be big, and you have to have, ideally you might start with a healthy but aging cohort, so maybe helping people in their 60s. But then finding the funding and the approval for all that is difficult, and that's something. Okay, so working in aging in humans is difficult, possible, and needs to be done. But to make it easy is that I work with a little nematode worm called C. elegans, which is much easier because they only live for about 2 or 3 weeks. And so you see the aging fast, and you can grow them really quickly. Their life cycle is every 3 days, and every 3 days that animal makes 300 new worms. So you can just get loads and loads of worms and do lots of studies really easily. This is a slide, I don't know if you can see that, maybe there's too much light on the screen, but you can basically see, put fluorescent proteins in them and see the organs in there. So you can see a fluorescence for all the neurons, and this is then particularly GABAergic neurons and cholinergic neurons. Actually, Sidney Brenner, when he set up, that's great, thanks. When Sidney Brenner set up the C. elegans as a model organism back in the late 60s, that's what he wanted it to do, was actually to understand the nervous system, because it only has 300 neurons, so the idea is that if we can understand the worm brain, we can understand our brain. And there's been a lot of research on that, and people working with C. elegans have won 4 Nobel Prizes, but we still don't understand how this 300 neuron brain really works. But that's another subject. But it has all sorts in there. It has intestines, excretory muscles, epidermis, epidermal cells, germline. It doesn't have things like eyes or legs, but that's their secondary things to worry about. It swims around. Anyway, a lot of the genes and things have very amazing homology to what we find in humans. So people have discovered a whole load of genes and things in C. elegans that have been useful for human research. And it's been good for aging because of mutants in C. elegans that were found. The first mutants that were ever found that made an animal live longer were found in C. elegans. They found mutants that made a worm live double its length or 50% longer. And that was kind of started the whole idea of the field of looking at molecular analysis of aging. And that's kind of where I got into it, because I was actually working on Drosophila melanogaster fruit fly. I was working on an enzyme called PI3 kinase. And while I was doing my PhD, it was found that the mutant of the worm that lived longer was also a mutation in PI3 kinase. And so that pathway, which is very conserved, seems to affect aging. And the downstream effect of that pathway was a gene called FOXO3A. You find variants of that in long-lived human groups. So there's definitely a link there. So, but my research has been to do, so yeah, this is some of the things that I've mentioned already. So my research has been to do with these worms and the food that we grow them on in the lab, which is E. coli. And so these worms, the thing about it is that in a way these worms eat the E. coli, and the E. coli grow from the medium, the agar that they grow on. And so so it's kind of like an analysis of the nutrient microbiome and the host. And what's good about the system is that you can control it, so you can chemically define what's in the agar. You can do genetics on the E. coli and drugs on the E. coli, and you can do the same genetics and drugs for the worms, so you can have good control of the whole system. In the wild, C. elegans is found all over the world, and it's found in rotting vegetation and rotting apples. They really love rotting apples. And they eat the bacteria that are growing on the rotting apple. And you can see from the electron micrograph that we did, you can see the bacteria inside the intestine, and you can see the microvilli from the intestinal cells. And so that's what they're trying to do, but they're surrounded by microbes, so they're a source of food, but also danger, because the microbes can be pathogenic and want to eat the worm. So that's what they have to live in this world, of food and pathogens and everything, right? So what we do this. So in a way, what's happening is the worm is taking the energy that originally was in the apple, but via the bacteria, and then eating it via the bacteria and using it to grow. What we do is we eat an apple, but instead of we can get a lot of the energy of the apple via bacteria in our guts. So instead of having to eat bacteria, we can carry it around, and it helps us get some of the nutrient out of our food. So, but we have a great immune system and a great intestinal barrier that stops the bacteria invading us and killing us off, right? So we walk around with this potential bag of pathogenic bacteria, but we keep them under control, and we use them to help our digestion. So, why I'm saying this was because I kind of by accident came across a mutation in the E. coli that made the worms live a lot longer. So I was working with this is a survival code. You start off with 100% worms alive and eventually they're all dead over a certain number of days. And this actually controls a long-lived mutant that lives longer than normal. And then I was feeding it bacteria in which you can put in a plasmid for a worm gene, and then by RNA interference, which is like small RNAs, it can knock out the worm gene. So I was looking at that to see if the genes that affected that and affected the longevity of this long-lived mutant. And the FOXO homolog DAF16, if you knock that out with RNAi, you make the worm live a lot shorter. But then I found a strain of E. coli that made the worms live a lot longer. And all right, even longer than the mutant that was already long-lived. So I thought, wow, this is a cool observation. It wasn't what I was expecting. it was there. I thought it was knocking out this gene UGC27 because it had the plasmid to do that. But it turned out that wasn't the case at all. It wasn't. That was a wrong assumption. It's a long story, but I worked out that it wasn't that knocking out that worm gene, because when I later retransformed that gene into plasmid to new E. coli, it didn't affect the worm. And when I got rid of the plasmid, the worm still lived longer. So it wasn't anything to do with the worm gene, it was the E. coli gene that had been mutated by accident, by chance. So quite a lot of work later, I found that doing some old school molecular biology, I found that gene was this gene called ARO-D. Now ARO-D is a gene that in this biochemical pathway that makes aromatic compounds. And so you've heard of like aromatic amino acids like phenylalanine and tyrosine and so on. But it turned out that the important one was para-aminobenzoic acid, which is a precursor of folates. And so we could do a lifespan, this is like where I said you could manipulate the media, we could have the long-lived mutant, but then we put para-aminobenzoic acid back into the media, then the worm was no longer long-lived. It had a normal lifespan. So it showed it was to do with, it proved that it was to do with this folate pathway. So what we were doing was stopping the bacteria producing folate, and then the worm lived longer. And we could do the same thing with an inhibitor. There's a sulfonamide drug called sulfamethoxazole, which inhibits bacterial folate synthesis. And that has a dose-dependent effect on the lifespan, and we can achieve over a 50% increase in lifespan if we slowly increase the amount of this drug that inhibits folate synthesis. So you might be thinking, well, this is just an antibiotic that kills the bacteria, but we find that we could get the effect at concentrations that didn't kill the bacteria, that still allowed the bacteria to grow. So the folate cycle, this is a very simplified version of the folate cycle, it allows growth for biosynthesis, it allows transfer of one carbon molecules to make things like pyrimidines and purines that make DNA and RNA, and some amino acids like serine and glycine and methionine, which are used for proteins, for other methylation, things like that. But it's like a cycle, it's an enzymatic cycle, so the folate gets recycled. So you actually don't need much folate for this to happen. So what we find in bacteria is that we can grow, when we give the drug, the bacteria still grows, even though we've given them the sulfonamide drug. Still enough folate around them to grow, so it's like a cycle, but it's a thinner wheel, but it's still turning. And then with the worm, this is a little video of the worm, you can bring down the folate levels in the worm if you treat the bacteria with SMX, because the worms depend on the bacteria for folate, but the worms are fine on this. They have the same growth rates, they have the same amount of progeny, so they can exist on a lot less folate than they're being exposed to. So they don't need that much folate. So the question is, why was it living longer? Was it to do with the folates, less folates in the E. coli or less folates in the C. elegans? And the way I like to think about these things is like, when you have this sort of question, you can think about a spectrum of possibilities, and then you try and eliminate different possibilities. So the one side, it could be that we're stopping the E. coli being invasively pathogenic or producing toxins, but maybe if we reduce the amount of folates or maybe something else to do with C. elegans metabolism, so the folates, then that's what's happening. We tried to deal with this by looking at a gene called GCP2, which we have in our intestine, which allows us it takes off glutamates from folate and allows us to take up folates. So we found a mutant in the worm, and we found a mutant. We got a mutant worm with this, and we found that that this mutant worm, which is called the GCP2.1 mutant, grows fine on normal E. coli, and normal wild-type worms grow fine on SMX-treated E. coli, but this mutant does not grow on E. coli that have the sulfonamide drug, so they're just more dependent on folate, those worms. So that means we could, so then one question was, well, okay, do these worms live longer because they're having less folate? And the answer was no, they don't live longer, if anything they live slightly shorter. And then we could use this system to see if we could rescue back that growth So we added folic acid, which is a supplement, and folinic acid, which is a reduced form of folic acid. It's not found, it's more like the natural folic, which is found in nature. The supplement that we have folic acid in, that we have in supplements that we give to people is an oxidized form that's not found in nature, but it's more stable and it's cheaper to make, because that's why we use it. And we could still use it, but the worm's not very good at taking it up. It will take it up eventually. But it meant that we could know that we could have conditions where we rescue this. So we supplemented the worm with folinic acid, and we still saw that the worm lived longer when we treated with SMX. So that shows it wasn't to do with the folates in the worm, it was to do with the folates in the bacteria. So kind of like simplified version of this. It's good to have folates in animals, but it's not necessarily good to have folates in bacteria. There's one thing you should like take away from that, is that that's what we think, you know. And so what we did was, we were thinking, well, okay, this mutant was found by accident, maybe there's other mutants, there's another one. So we decided, you know, the question was, if we found the other mutants, would it tell us what's going on, and how lucky were we to find this mutant, basically. And it's just about bacterial growth. So basically we did a screening of 1,000 E. coli mutants. And that's what you can do with something like C. elegans. You can put, for each strain of bacteria, we could put 100 worms on that strain of bacteria. So if you're trying to think about doing that within the other system, it's impossible, right? It did take us a lot of work, you know, it wasn't easy, but we did it, and we retested and did a full screen. And we found 9 genes, mutants that made the worms live longer. And 2 of them were PAB-A and PAB-B, which was needed to make PABA, which is good because that fitted what we found before. So if we had a mutant in PAB-A or PAB-B, they live longer. We put the gene back into the E. coli, they live shorter again. And that's the pathway that's used to make PABA. So, but it was the folate synthesis and not the folate cycle of the bacteria. So we didn't understand why that was. So our kind of hypothesis was that you need a low level of folate to support growth, but actually the E. coli is making much more folate than it needs for growth. And if we can stop that folate production, then it's less toxic. The E. coli is less toxic to the worms, and it's making them live longer. And we can manipulate the media with this mutant that we had, and we can add PABA into the media, and with lower levels of PABA, the worms live longer, high level PABA, they live shorter. So we can manipulate this really carefully and allow us to understand how this works. And our kind of hypothesis was that, okay, folate synthesis is needed for the folate cycle and growth, but that excess folate is somehow involved in other genes that we don't understand yet, and there's some sort of toxic activity that's causing lifespan decrease also makes the worms not like this bacteria, which is interesting. So is this relevant to humans? Is that folate synthesis bacteria associated with disease? So one thing we looked at was, we had a collaboration in China with Liping Xiaotong, and they found microbiome. Well, this isn't their work, but there's work in microbiome that if you take the microbiome from an genetically obese mouse and put it into a germ-free mouse, then that mouse becomes obese. And it doesn't happen from the control bacteria. So my collaborator Liping Zhao took a cohort of obese Chinese people, put them onto a low-calorie, high-carbohydrate diet, and looked at what happened to their microbiome. And he found that particular bacteria, proteobacteria, which is kind of a similar family as E. coli, would disappear from the gut of those people. And so he took one strain from an obese human, put it into a germ-free mouse, and on a high-fat diet, it made the mouse obese. So this had one particular strain of bacteria that could make a mouse obese. And so what we did, we set up a little lab in his lab in China where we grew our worms. We took our expertise over there, set up a little worm lab in China, and we grew this bacteria that he had from this obese human, and we found that the worms lived shorter on this bacteria. But also the worms didn't like this bacteria, and also they were kind of sterile. They didn't reproduce well this bacteria. But when we inhibited folate synthesis in that bacteria with SMX, all those things disappeared. The lifespan increased, they liked the bacteria, their fertility went up. So it fit with the idea that this bacteria is making lots of folate, this was causing some bad things happen to the animal, potentially causing the obesity in humans, but we could stop it by inhibiting folate synthesis. So we were kind of excited by this. We actually, at one point, we applied for a patent to look into this, but the patent office found this paper from 1958 in Germany from the Bayer Corporation, which were looking at people that actually made sulfonamides, and they were looking at sulfonamides to treat cancer. And they found that the sulfonamide drug made these mice, rats, and dogs all live a lot longer. It was a bit of an anecdotal paper, but it was really kind of intriguing, you know. And they put PABA back into the food, and they reversed that effect. And they didn't think it was through the bacteria, they thought it was going through the animal. What we maybe now would say is to do with the bacteria in the animal. And then I just really love this picture because this is a picture of mouse 99 taken on the 11th of December 1957. No, taken on the 12th of December 1957. And this is the same mouse after treatment on the 14th of January 1958, and it kind of looks a lot better. So I don't know how much to go on, but I like this picture. The lighting is different and everything. So we think that maybe inhibiting bacterial photosynthesis might be useful for suppressing obesity and slowing aging. One thing that is also known is that there's this condition SIBO, which is small intestinal bowel overgrowth. There's a clinical sign for it is increased serum folate levels. So maybe all those bacteria in the small intestine are producing a lot of folate that's getting into the serum, but maybe if we inhibit the folate, that could be useful for that. And you know, proteobacteria are associated with lots of chronic diseases. And as you get older, you know, you're much more likely to have like issues with gut barrier problems and that kind of thing, and more likely to have issues with bacteria. So your gut is no longer, your immune system, so your gut is no longer able to contain those bacteria. And so maybe we should worry about folate in those bacteria leading to toxicity of the bacteria. So that kind of made us think about folic acid supplementation, because in many countries folic acid is put into flour to prevent neural tube defects. And they're about to do that in the UK in next year. But what I'm worried about is that, okay, in the UK there's about 200 neural tube defects births a year, and we should, with that supplementation, we expect to prevent a maximum of 100 of those births. But we're giving folate to millions of people, many that aren't the target. The target area are women, you know, often in deprived areas who don't get enough folate in their diet, but there's many people that are getting that folate, pretty much older people. So they're kind of worried about that. And so we kind of did some work on this, and we found that, remember I told you that C. elegans wasn't very good with folic acid, but was much better with folinic acid. We found that what was going on was that the folic acid was having this breakdown product called PABA glue, where it kind of split here, and then that was taken up by this pathway in C. elegans, then turned into PABA, then resynthesized into folate, then the worm would take up the bacteria from the folate from the bacteria, not directly from the folic acid. And so this breakdown product can feed the bacteria, but it's not used. Humans can't use that breakdown product to make folate. Only the bacteria can use the breakdown product. So we took folic acid supplement from Boots, the chemist that plays in, and we measured how much of this breakdown product was, and there was like 3 to 4% of this folic acid was already had this breakdown product. And no one knew about this, and it's not considered when people are thinking about this supplementation. And the other thing was, what happens when you put it in flour and bake it? You're likely to cause more breakdown, you know, because heat breaks down. I was looking at this in the US. They do this, and I was looking at this salty snack that I was eating on an American airline, and they had folic acid in there. And what does that make in this snack? I'm sure that's going to break the folic acid down further, and you're going to have more of this breakdown product. So we're going to investigate that. Actually, we'd like to investigate that. We're also we're talking with the people that do the Twins UK study in King's College London to try and, they've got a study of 1,000 twins and looking at how they, as they get the supplementation in 2026, when everyone has it from the flour, how that affects their health and so on. So kind of like our idea is that, you know, dietary folate is good for you because you need folate. I'm not saying people don't need folate, you know, or worms don't need folate, but there's also folic acid can be taken to the breakdown product, taken to the E. coli, and although that also produces folate, that's good for us, it also produces what we think is like metabolites and activities that are bad for us. So we think that what we're trying to investigate is whether the same thing's happening in humans as well. And that maybe the microbiome is taking up that breakdown product and making things that might be bad for us. So there's a review on that. So that's kind of a summary of what we think is that mild inhibition of photosynthesis improves health and slows aging in our model and in the model with a bacteria from a human. So we want to investigate whether they might work as a therapy. Maybe some natural products already inhibit photosynthesis, and we want to find out what's going on with the breakdown products, especially in older people. I'm just going to show you this. How are we doing for time? Running out of time a little bit here. But I haven't got too many slides left, you'll be pleased to know. 5 minutes, yeah. So this is a technology developed by a company, Magnitude Biosciences. We're not just looking at lifespan, we're looking at health span. So we see how well the worms do with time, and we can track their motion. So basically we're looking at health by through motion. And we see that after a few days, the motion of the worms really plummets. But this drug that we tested for this company in California makes the worms stay healthy for much longer. So we work with companies. We've worked with like, you know, well over 50 companies, done 100 projects across 12 different countries, of people looking at drugs and supplements and things like that to see how they affect health. So that's kind of what we do there.
There's one worm on the controller.
Still going, isn't it? Still going. That's what I say. The variation. Variation. There's always someone that's still going. You know, that's why you get these people that live to 100.
Didn't do too bad. 1, 2, 3. That's surviving on that side.
Still going, aren't they? Yeah. And that's why you need large numbers and you need to do something, because there's always, you know. So when we did this with, so this is the lifespan with the SMX drug I was talking about. And we see this is actually quite low concentration. We see a dose-dependent effect. But when we look at our technology, that experiment took like 40 days. Whereas we use our technology, we see this decline of movement within a week, within 7 days. And so we can just speed things along. We don't have to do these long lifespans, which might not be long, but if you're going through thousands of conditions, it's long. We can now do it within a week, you know, so that's what we're trying to do. But also shows you that it improves the health span for animals as well as the lifespan. So what I'd like to do in those, use these E. coli health span assays and then trying to do some fractionation of what's coming out of the E. coli to find the molecules that are causing the aging and try and understand the aging at the molecular level. Right, so now I'm going to switch back to something that's a bit of a paradox. So I was just telling you about how we can how we can use C. elegans to understand aging in humans, and it kind of doesn't make sense, because how could the aging of a thing that lives for 3 weeks tell you anything about aging of something that lives for 80 years, you know? But it does seem to work, and it works across different things, so why does that work, you know? And I think it's because the things that we found so far seem very, very fundamental and conserved. You know, things we found, like this insulin PI3 kinase system is found in worms and flies and humans, mice, it all seems to affect aging. There's another pathway, the TOR pathway, which is a drug called rapamycin inhibits it. You see that in yeast, it inhibits TOR and it affects aging. It still seems to work. So, and calorie restriction, if we restrict the diet of, we don't know if it works in humans yet, but it does improve health in humans, definitely. We don't know if it affects lifespan, but it definitely works in many organisms, in mice and many other animals. It works. So how does that happen? And I think that what I've just talked to you about, bacteria-animal interaction, is a very, you know, we evolved always having to deal with bacteria. We evolved into a world that was full of bacteria. So animal bacteria is also a very fundamental thing that I think can affect aging. So why are these, ignore that, why does that happen? Well, I haven't really got time to go through it, but there's this theory that we can talk about later called the interdependent network theory of model, which basically shows that kind of models an organism as a network that depends on other different nodes. And if you do this mathematically, I won't go through it, you can see that this mathematical model of a network collapses in the same way that we see that with animals, and we can map the mortality curves to worms and flies all sorts of things, and we can fit that model to what we see in nature. So that kind of says that aging isn't anything like special really, it's just a property of a complex interdependent network. And so another way I think about this is like aging is kind of like in circles and not in straight lines. You know, it's like people often think of aging as like, okay, oxidation of DNA damage, cell senescence, tissue decline, organ decline, aging. And that's not saying that's not true, but you know, if you have organ decline, it can lead to oxidative damage, so you get a circle that goes back. So you have all these circles of dependencies. And so that means that if you intervene in one type of aging, you can actually slow aging. So I think when someone has a better diet or they treat their blood pressure, you actually are slowing aging. And we see that aging is slowing because people are living longer and staying healthy for longer. So I think we are slowing aging. It's not like it's just finding better ways to do it. And you know, blood pressure is a great one. I won't go into that because of time. But then, you know, they have this thing that I've mentioned before, treating healthy people is a much higher safety barrier than treating someone with a disease. But that's kind of like where a lot of what we do is, but it might be too late to intervene in that kind of thing. So that's just something to think about, as what I've talked about already. But I do think as well that, you know, basic scientists have to have a more translational mindset. And even though I'm a basic scientist and I very strongly support basic scientists must always be thinking about how can we just get this new discovery translated. It's not just, you know, basic scientists are often, they just want to get the grants, publish the papers, because that's what helps them in their career. But actually, we want to actually make a difference, you know, and it's not that easy. You can, what happens is a basic scientist ends up guessing how their basic science is going to be useful. They don't actually know how it's going to be useful. So the way to find out if your technology is going to be useful is to talk to people who actually can use the technology. So this is from a talk that I had before, but I think it might still be relevant. You know, like I've got this kind of, someone said, you didn't have a call to action in your talk, so here's a call to action, right? So for the basic scientists, I think they should still keep investing. I don't know if there's any basic, people who call themselves basic scientists in the room. So keep investigating the effects of aging or whatever it is you're working on. But be careful about exaggerating the impact of your research, because we all fall into that trap. Reach out to those in prevention of disease, including clinicians, industry, charity, and think creatively about possible uses of your research, which is what we call in the business world a value proposition. And then you have to test that value proposition by actually talking to the end users to see if it's real. You know? And if it is positive, get that funding and get investors and go for it. So I changed this one because I thought this is called as more disease-specific translation research, right? So if you are working in a specific therapeutic area, which I think probably many of you are, I think you should look at where your intervention acts in the progression of disease, like I was talking about from the early disease to the late disease, and how much prevention is involved. And then make sure if you're doing any preclinical or clinical trials that have to do with aging, you've got to make sure your trials are long enough and broad enough to actually see the effects on aging. And share your challenge, talk to the other way around, talk to the basic scientists that are working on aging and share what you're trying to do, and I'm sure they'll come up with some good ideas to help you. And then we also have to collaborate across sectors. So what I'm trying to do in my role as chair of British Society Research on Aging, because I've also had interaction with the health and supplement industry. I'm a judge on the nutrient ingredients awards for healthy aging, for example. It's like bringing all these sectors together. Everyone wants prevention of disease, but it's very hard for each funding body to do it on their own because of the challenges. So the charity sector, the clinical sector, the government sector, the industry all have to work together. You know? This is what we're trying to do in BSRA. We've got these 5 pillars of connecting scientists, engaging with the public, advocating for aging research, funding research, and translating research, basically. So that's what we're trying to do. So, just to wrap up, my conclusions are, well, the problem of aging and the challenge of aging research, I haven't really told. There is a problem, I just tried to show you what it is. I think that working with the prevention of disease is really important and something I'm trying to build in different parts of the UK, but maybe in parts of the world. Let's try and work on that. I like to think about aging as disruption of the network, which I can talk about further later if anyone's interested in. And then I hopefully showed you that my own research with C. elegans showed that animal-bacterial interactions may be fundamental and influence human aging, so we need to investigate a bit more. A colleague of mine also wanted me to tell that if anyone wants if any of your students are interested in the Masters in Biotechnology, we have it at Durham University, and we have students from all over the world there, and they can do projects, like real proper projects in labs, as well as learn about industry and all that kind of stuff. So they have a 6-month research project. So I'll put their slides up, but if you're interested, come talk to me. These are all the people that helped me with my survey about biology of aging in the UK from all over, also not only the UK, all over the world. And these are the people that helped me for my research. So that's it.
That's absolutely fascinating. Really exciting. So we're going to open this up for questions, but as the chair, I have the first prerogative. So a couple of, so you showed that slide about aging, with people living in more deprived situations as compared to people who are more affluent.
Yeah.
And I was wondering, the shorter lifespan. Is it those people are more likely to be doing manual labor, working manually, and maybe they also have a lot of injuries from working in that area. And obviously, the more affluent people have more sedentary, good nutritious life, and so it kind of that may be a factor to that. Why people in those areas it might be the way they work. So that's my first question. My second question is that so you used E. coli, the proletariat of the lab, as your substrate, and in the Shanghai lab you used a different substrate. So my question is, you have this lovely experiment, and what happens if you change the substrate like you did in Shanghai? Try to find, would changing the substrate also help you, uncover some mechanisms? So
No, definitely, yeah.
So
So just answer that one first, and I'll go back to the one.
yeah.
So that's the beauty of this. That's why we can vote those different mutants in E. coli. So we brought it down to not just changing between species, but we could actually have the same organism, but just different genes knocked out. There's a library of mutants from Japan, where you have a Kyo collection where each individual gene is knocked out. So that's what we were doing to try and understand that. If you go from E. coli to that into back to cloaca, even though they're quite closely related, there will be hundreds of genes that are different between those two. So it's harder to really pin it down, but possible, yeah. So there's so much potential from understanding the system, but still a lot of work. What I was amazed about E. coli, because like you say, E. coli, we all work with E. coli in the lab. E. coli is probably the best studied organism that we have, right? But E. coli has 4,000 genes, and almost a third of them, or more than a third of them, we have no idea what they do. We just still don't understand what they do. So there's so much we don't understand about just E. coli. But this is a system that can help us understand it. The reason why we probably don't understand it is we don't have the system where those genes are probably really important for the E. coli inside the organism, right? But we don't look at it like that. We look at it in a flask. So we need to get it in the situations where those genes are important. So that's that. The deprived area versus non-deprived. In the UK, yes, there's people doing manual labour, but there's also a lot of other things. Like as people are more deprived, they're more likely to have like alcohol misuse, smoking, drug use, poor diet, like you said, maybe doing less exercise. So it's very complex. There's a lot of different things. But what people have found that if you look at life expectancy and income, it goes up, and even like something in America where you can look very, very wealthy people, it still keeps going up. So there's some connection there. It's probably, from my way of thinking about it, the network theory. When you've got more money, you've got more things to fall back on. So if something goes wrong, you've got things to fall back on. So it's like your network is stronger, and you can buy that. So if you're living in a very low income, then you're always on the edge. There's always problems, and they can just get out of control. And so I don't know, but still, there's a lot to understand there.
So my last question, and then you can have a chance. What about sulfur-containing drugs, you know what I'm saying?
Yeah.
As a microdosing, so as a preventive strategy, so what happens if you had that as a prevention strategy to slow? Would that work?
The problem is that, well, first of all, the sulfonamides they use antibiotics, and when they use it at quite high concentrations, not actually very good antibiotics. And they've got reported toxicities at those high doses. It might be that at microdoses you don't have any toxicity, and then there's also the issue of getting selected for resistance, and that's another issue. So, but maybe it would work, but the problem is they will never let you do it.
Okay.
But yeah.
That's fascinating.
Yeah, this is the hypothesis.
So let's, coming from your perspective, what are the kind of things that we could leverage? So I found that the issue of the in taking someone who is obese, that experiment. How do we apply? How can we do some of this kind of work at Wits University?
So, a disclaimer, I'm a basic scientist, and we had the School of Biomedical Sciences annual research day yesterday. And our head of school got very, very offended by being called a basic scientist. He's like, this misnomer of being a basic scientist, because the clinicians in our faculty always think that the basic science is a little bit dumb. But it's, I'm just being frivolous. But I do agree that in terms of, and I come from a background of, actually I started out as an exercise physiologist, and I work with humans and human health and exercise and whatever. And now I have a molecular lab. And so I came backwards from population sciences into, and one of the things I always tell my students is like, what does this matter to the people in your audience who doesn't care about the down to the molecular level? And so I think that comment that you made, like, how do you make your science translatable? And I think what we've been trying to do with IDORI really is to get that network of both clinicians, basic scientists, pharmacists, and actually be able to say, this is where I can actually contribute. So I think this is really important for our faculty, because we have got a very siloed approach. We sit in our lab on the 6th floor and nobody knows that we exist. So
you have to get out of that.
Yes, exactly. So I think we are busy building like sort of a multidisciplinary science park, really, that where we are trying to get people to actually just talk to each other, because we've got a very, very rich resource of patient samples, population data, biomolecular kind of work, and then implementation sciences. But I mean, these things take long. The pipeline is so long.
So one thing I would say, so I learned about this a lot through working with my spin-out company. Because what you need to do, if you want to get someone interested in your research, you've got to understand what their challenges are, what their problems, what's their biggest barrier to getting where they want to be. And then once you understand their challenge, then you can see whether or not you can adapt your research or use which could be useful in their challenge. But if you just talk about it like, oh, this is interesting, it's not going to get there until they see it actually being useful. And that, you have to have a lot of conversations that way. You have to have a lot of conversations, really, like, not just, oh, hi, you have to kind of really understand what they're trying to do. And then you can try and work out where you can use your research. And then you have to think about then you might have a crazy idea like I've got here with the photos. Then you've got to say, well, what's the next step that makes it more like, like a de-risking step? Like, how do you make that idea has got potential impact but a lot of risk that it might not work? What's the next step to show that that's more likely to work? And then work like that and work along that pathway. But basic scientists don't often think like that. They think, oh, let's find the next cool molecule and then we can publish it. And that's fine, that's all good. But if you've got a choice of a few different molecules, if there's one that might be more useful than another to someone, then go with that one. You know what I mean? Like, that's why just the way you're always making choices about what particular direction you take. But if you've got someone that you talk to that's got a particular thing they want to do, then you might as well go in that direction. So it's having those conversations.
So Xavier, you're going to go to Agincourt now, and they have this demographic platform, and they've been watching this population over the last 25 years? 30 years. Okay, coming for 5 years. But so there you have a wonderful demographic platform that is watching aging unfold. And what you've got, and maybe I'm over speaking what you've got. Besides the data, but you don't have dry blood spots and data on diet. You know, so could one leverage? Okay, so you've got this aging cohort, and obviously a lot of things have changed. ARVs came, you know, Asian cohorts got wealthier over time. The basic income grant, and there are a lot of things that could have helped with the longevity. But you also have, you know, besides watching them age and the kind of structural changes that help them, you also have, you know, biological data. So you could look at, you know, you could do a case control almost. You know, do a case control. You know, those without HIV, blah blah blah, who match them. So you have your twin, your digital twin, and you basically match them, and then you've got your dry blood spots, and maybe you've got diet history as well, which maybe I mean, so when you go there, you might want to think that. But maybe you want to just talk about some of your aging work that you do.
Yeah, well, certainly.
Yeah. Last question, then we'll get some food.
Let's change our microbiome.
It's good to start.
As we said before, we have seen the changes in this population. The population is aging. Since 2010, we have started collecting all these biological samples, and microbiome is one of them. We are starting now. I mean, it was fascinating to hear all that, because I was thinking how we can explore with this microbiome and with other blood that we have, any other IDORI, we have other things. I don't know how much we can use, but it's to explore what can we find into these samples that can also explain this issue of aging. And for example, you were saying in the UK, people that are poor or low socioeconomic status, they live less years of healthy life. What is in our sites, by definition, they will be lower standards than what they have poor there. Then when do you consider a person to be an old person? Is the same in the UK or in a rich environment than in this rural area? And what makes people get older? And why another thing that we have seen, and it's all over the world, is like women have longer lives, but also with longer time of terrible life. You know, it's like, I mean, it's not that you are living healthier, live longer. Then all these things can also be jungle. I mean, we can discuss more, and we have to discuss more what can be done there. Yeah, sure.
So first of all, thank you very much, and thank you for bringing your father. We do like to have some networking, and so we're going to go across the way. We believe that alcohol, wine, especially, increases the lifespan. That's how I justify it as a medicine for all of us. So please come along, please come and join us for food. Thanks.